At CBAF, scientists can advance our understanding of matter itself by probing the quarks and gluons in the atom's nucleus with beams of electrons from the superconducting accelerator. CBAF has an educational mission as well. The CBAF Science Series features scientists and engineers who are gifted at making their work not only informative, but fun. The Science Series is held once a month during the school year in the CBAF Center Auditorium. The presentation is live, spontaneous, and for the most part, unrehearsed.
Good evening, everyone. I am Kathy Strozak, CBAF's Education Program Coordinator. We at the Continuous Electron Beam Accelerator Facility... Should we start again? Let's start again. Good evening, everyone, and welcome. I'm Kathy Strozak, CBAF's Education Program Coordinator. We at the Continuous Electron Beam Accelerator Facility appreciate your patronage of our science series, which is now entering its 4th year. And we thank you for making it such a success. And I hope tonight you enjoy our presentation, Jellyfish, the Big Sting, by Dr. Karen Rowe. Dr. Rowe is an assistant professor at Hampton University. She earned her doctorate at the University of Maryland in Baltimore and her master's degree in the College of Marine Studies at the University of Delaware. Since high school, she has had an interest in studying venomous marine animals. But hey, haven't we all? In her free time, Dr. Rowe enjoys scuba diving, an interest she also acquired in high school, this time from an astronomy instructor. I think there's a little bit of vertigo here, but let us hope that she never encounters venomous marine animals while scuba diving. Anyway, thank you so much for attending and enjoy tonight's presentation.
Don't forget to turn me on. Thank you, Kathy. Good evening, everybody. I think I'm happy to be here, but it truly is an honor and a privilege to come out and be able to speak to you all here at CBAF. May we have the first slide, please? Woo. Okay, well, the aspects of jellyfish research is a large variety of different types of research aspects that anybody could be involved with. Okay, jellyfish are in the phylum of cnidarians, which is mainly marine invertebrate organisms. Another types of cnidarians that are also present are the freshwater hydras, sea anemones, and corals that are all relatives of jellyfish. Okay, this slide illustrates some jellyfish, 3 out of the 4 classes of cnidarians. The bottom one here is a representative sample of the Scyphozoan class, which are the true jellyfish. They are characterized by having a bowl-shaped bill with scallop margins on them. Local species that are found here in the Chesapeake Bay include the lion's mane's jellyfish, Cyanea capillata, the moon jellyfish, Aurelia aurita, and the lovely sea nettle that you might have met before, which is Chrysiara quinquecera. A lot of these jellyfish that are found locally, such as the Scyphozoan jellyfish, will produce no detectable to maybe like a moderate sting. And for those of you who have gotten stung by a sea nettle before, you may not consider that a moderate sting, but it is in comparison to some of the other jellyfish that you might encounter. The second type of jellyfish is a representative sample from the class Cubozoa. These are the most venomous types of jellyfish that you'd ever want to run into. They're characterized by having 4 flattened sides on the bill with 4 tentacle clusters or 4 tentacles on each of the corners of the flattened sides of the bill. Representative samples that I don't know if you have heard about them before, maybe if you've traveled to Australia, is the notorious Australian sea wasp, Chironex fleckeri. Other box jellyfish that are also relatives of it include Charybdia and Chiropsalmus. These species are found in subtropical and tropical oceans and they are responsible for producing at least 2 deaths annually by people who have gotten stung severely enough by them. The third class of cnidarians is the hydrozoans. This representative sample here is called Physalia physalis, better known as the blue bottle or the Portuguese man-of-war. It's not a true jellyfish as observed in this scyphozoan and the cubozoan jellyfish here, because within their bell margins they're composed of a gelatinous type of material. This jellyfish here has a float that stays on the surface. There's no jelly inside that float. It contains like mostly 90% of carbon monoxide gas, and it is really composed of a bunch of specialized polyps and medusa that's a colony of organisms that functions as one organism. So with the carbon monoxide in the float, up here it stays at the air-water interface all the time, and its tentacles may trail several meters below the surface of the water. Okay, these are 2 samples of another hydrozoan type of jellyfish. These are the more likely ones that you'd run into anywhere around. They're not colonies, they're true jellyfish. The genus name for this is Gonionemus, and they're very small. They're about the size of an eraser head on a pencil. Whoops. Okay, whoops. Okay, the last class.
Could we have the lights a little so we
Yeah, oh, okay. Do I have that, or do you have that?
This is the channel 6 lights that are on the screen. I can cut our lights completely off.
Okay, well, this is a 4th class in the cnidarian phylum. It is called the Anthozoa, and it includes the corals and the sea anemones. The difference between this and the other groups that I just talked about, or the other classes of jellyfish, it never exhibits like a free-swimming medusoid stage. Rather, they stay like as sessile polyps for all of their lifetime. And a lot of the classification of cnidarians is dependent on whether or not these organisms spend most of their time as a sessile or attached polyp stage, or if it stays like a free-swimming medusa stage, and then certain animals travel through both cycles somewhere along their life cycle. And that also tells you a lot to do about why you get such a massive bloom of jellyfish sometimes during the summer, because of this alternation of generations. That's what it's called. This is a representative sample of Aurelia aorta, but the sea nettle has a similar type of life cycle. It starts out as an adult medusa, they think. There's a big question about which came first, the medusa or the polyp, just like what came first, the chicken or the egg. But so we'll say that the medusa started out first. And medusa have separate sexes, which means that they're gonochoristic or dioecious, which is terms for that. So they produce either sperm or eggs, and the fertilized eggs turn into a larval stage called a planula. The planula is ciliated, and it swims for a while, and then it settles down to the bottom and attaches onto something, and it begins to grow at the beginning of its polyp stage, where it's called a scyphostoma. And then the scyphostoma can start to grow some more, and then through a process called transverse fission, it starts forming a plate of stack-like structures. I think I said that backwards. Okay, a stack of plate-like structures, and now it's called a strobila. And what happens after this, the strobila buds off these plate structures, which are young ephyra, okay, and then they mature again and turn into the adult medusa. So the alternation of generations comes from the sexual process of the medusa to the polyp stage, and then you have asexual reproduction from the polyp back up to the medusa stage. So this is why you can get a nice big massive bloom of jellyfish during the summer when the temperature and salinity is correct. And also, after all of these ephyra bud off of the strobila or the scyphostoma does not necessarily die. What it does, it can go dormant, for over a year or over several years. And again, as the temperature warms up and things get nice during the spring and summer, it can start strobilating, I hope that's a word, making all these stack of young euphyra again, and then those bud off. Additionally, what's not shown on here is a scyphostoma can also branch out laterally and form extra buds on there, so then you can have strobilus branching out from 1, 2, or 3 different places on there. So, massive jellyfish blooms. Okay, you can't see this though. This was a computer-generated image of a jellyfish, and a lot of people think that there is no economical value to jellyfish other than having them being served as a dried or marinated Chinese food. But within the last decade, a lot of researchers internationally have found that, that jellyfish may be top predators on a lot of the zooplankton, and they might tend to crop down the population, so they have a certain amount of ecological importance in the environment. Okay, this is the moon jellyfish, Aurelia aorta, and a study was conducted out in the western Baltic Sea on Aurelia aorta, and they found that these medusae caused a reduction in copepod populations. Copepods are small crustacean-like animals. They're like little teeny shrimp. If you've ever drank some seawater out in the ocean someplace, you probably swallowed a bunch of them. Okay, so you're taking somebody's food away. Don't do that. Okay, and so they expected them to reduce these. They know that jellyfish are carnivores and they're going to eat these crustaceans. But also what they found is that they reduced the yolk sac herring larvae. And so I'm sure a lot of commercial fishermen are upset about that, not only because these are also food sources for other things, but they can also be used for human food source as well. Aside from that, the other important thing is that copepods are phytophagous, so they just eat plain dinoflagellates and diatoms and things like that. So they're the primary producers. First is the phytoplankton, then comes these. So these are abundant, and they're a big food source for a lot of things. But this yolk sac herring larva is another trophic level up away from the copepods. So this moon jellyfish is like tapping into more than one trophic level. And so that can pretty much screw up the ecology in some cases. Again, another scyphozoan jellyfish, Pelagia noctiluca. A study was done in the Adriatic Sea on this jellyfish, and they analyzed the contents of the gastrovascular cavity. And that's just like saying they did a stomach content analysis. And so what they found is that Pelagia medusa consumed primarily copepods and cladocerans. For any crustacean type people, this right here is not a cladoceran, it's a fairy shrimp. But if they were out in the water, they'd eat them too. Secondarily, chaetognaths, and these are planktonic arrowworms that are abundant in the plankton. And the third thing that they found that Pelagia ate the most was mollusk and teleost eggs and larvae. Mollusks being organisms like clams and snails and octopods and squid are all part of the mollusk phylum, and teleost means bony or spiny fishes. Okay, so the footnote to this here study is that they found that the relative abundance of these major phytoplankton, zooplankton that are found in the column right here were the ones that were picked off the fastest. So they decided that Pelagia does not have any type of selective feeding behavior, and what will happen is they will tend to just grab up anything that's in the water column at the time of their bloom, so they will tend to deplete the most dominant forms when they're growing up. And so that'll have a big ecological impact as well, because the more dominant forms will become less dominant. Okay, well, this is a picture of a sea nettle. And just some basic external morphological features of it. Again, the bell with the gelatinous type substance in it. These tentacles on the outside perimeter are the fishing tentacles. These are the ones that will sting you the most. And then there's mesenteric tentacles that are found on the underside of the bell as well at the periphery of it. And they encircle the mouth or the manubrium. Regardless of how you transversely slice any cnidarian, just not jellyfish, but hydras or sea anemones or corals or anything, they all have about the same basic body plan. They'll have an epidermal surface layer, and they'll have an underlying gastrodermis layer that lies within the gastrovascular cavity. or the stomach area, where digestion occurs. In between the epidermis and the gastrodermis is a layer called the mesoglea, which is a fibrous mucoid layer that's basically acellular. There might be a few cells in it depending on the species of it. But since they don't have like a real third cell layer in between there, cnidarians are considered, or they're characterized as being diploblastic, which means 2 germ cell layers, as opposed to higher organisms that are triploblastic, where they'd have like the 3 cell layers. Okay, so they have an epidermis, a gastrodermis, and this mesoglea layer is right here. And you might think it's jelly, but it's not. Okay? All right, so on the epidermal layer right here, there's 5 basic cell types that are present. And there's about 5 basic cell types present on the gastrodermis as well. They're pretty much similar. There are a few differences in them. In the epidermal layer, first important type of cell is epitheliomuscular cells. These are columnar-shaped cells or rectangular-shaped cells, and their bases are slightly extended. The extended part of the cells helps to form the epithelial surface on them. And they also have muscle fibers going through them, which helps with the elasticity and flexibility of the jellyfish during its movement. Okay, the second type of cell, they don't have it listed on here, but it's this round one. It's a little round cell with the big round nucleus in it right here, and these are called interstitial cells. Interstitial cells give rise to the sperm and the eggs, as well as to any other cells that are needed within the body of the jellyfish. And cells a lot within vertebrates that have this specialized function or the ability to transform or give rise to any other cell type is needed, the term is used as totipotent. Okay, so the interstitial cells here are the totipotent cells of the jellyfish. Okay, third type of cell that's in there are the mucus secreting cells. This diagram doesn't show them up here, but they look similar to the epitheliomuscular cell in their morphology, and they make the jellyfish slimy, basically, I guess. From the outside, the inside may have some food digestive function. Okay, the fourth type of cell, which is really 2 types, are the receptor cells and the neurons. Okay, the receptor cells are particularly abundant on the tentacles of the jellyfish. It helps it in tactile function. Okay, and they're in close association with the neurons that probably lead to the nerve ring. Jellyfish don't have brains. They just have like a nerve plexus that encircles around the outer perimeter of the bell. Okay, and the fifth type of cell that is the most interesting cell is called the cnidocyte cell, because it contains organelles called nematocysts, and these are what stings you. Okay, the nematocysts are double-walled capsules made out of a collagenous-like type of material, and they've got like a tightly coiled and inverted tubule inside, and upon appropriate mechanical and chemical stimulation, the tube is thrust out of the capsule and shot into the victim. In some cases, some of these nematocyst capsules have barbs and spines on them. Okay, and there's an array of different types also. There's big ones and little ones. A couple of these have been fired already. This is not directly taken off of the tentacle itself. These have been isolated away from the jellyfish for other purposes. Okay, and all right, there's an array of different types of nematocysts. These are just some of them. A lot of them are difficult to characterize because they're either broken or you don't get them all in the same pieces. But in general, there's 3 basic types of nematocysts that you can get in any one species of jellyfish or any kind of cnidarian. And commonly what you'll find is they'll have more than one type of nematocyst, even in one species. So this is going to be an old terminology that I tell you about. The first type are the volvents. Volvents are the nematocysts that are unarmed. They're closed at the tip, okay, and they're coiled upon discharge. So they would probably just function in wrapping around prey for whatever reason. The second type of nematocysts are the penetrants. And these are the ones that will sting you and inject a toxin of proteins and phenols and anything else that they haven't identified in the toxin as of yet. This is one of them down here. Okay, and they have like open tips on them also. Some of these, they're all open tips. They've got spines on them. They act like as little tiny harpoons or syringes to inject toxin into the victim or prey item. The third type of nematocysts are the glutenants, and these are opened at the ends also. It doesn't even look like that here, but they are. They're open at the ends. They don't have any toxin in them, but they have like sticky substances in them, and they will help to anchor the animal under certain conditions if it needs to be anchored, or they might be used to help hold a prey item so the animal can sting it with its penetrance or something. Okay. Now, the $64,000 question is, what causes a nematocyst to discharge? There's a couple of hypotheses that they have thought about and they have hypothesized about, and it was based on the observation that before the nematocyst discharges its tubule out of the capsule changes in volume. So first it swells up, and then after the tubule everts out of the capsule, then the capsule shrinks the rest of the way. And the first theory is the osmotic theory, where they felt that some type of mechanism is going to cause the permeability of the wall. Remember, this is a double-walled capsule in some cases. Change the permeability of the wall to increase towards water molecules. And then water molecules would then rush in, and then the tubule would jump out and evert, and then get stung, right? Okay, they had to modify this because they found later on that the nematocyst capsule, even at rest, is freely permeable to water molecules and other larger molecules as well. So there's all kinds of things going in and out of here. So for diffusion mechanisms, there was nothing to make water rush into the capsule. To compound matters also, they also had observed that free calcium ions leave from out of the nematocyst capsule before the tubule discharges. So then they had to modify this and say, okay, well, maybe free calcium runs out of the molecules, and it was inside the cell before, and it was aggregating all of these proteins, but then it let go of them, and then all of these proteins dissociated, and then that caused extra water molecules to get recalled into the tubule, and then the tubule came out and everted, and you got stung. Okay, so if you don't buy that theory, there's at least another one. And this one is the constant pressure theory. The constant pressure theory states that they have this coiled up tubule inside this capsule right here, along with this proteinaceous fluid, and everything is just like swelling and bulging, just waiting to come out. And through some unknown mechanism, this little cap here, you can see it better on this side, this opercular cap flips open, and because of all of that pressure that's inside there, through spring action, the tubule everts out, and you get stung. Okay, but so since they know that like the water molecules are freely permeable in and out of the capsule, they couldn't maintain the same constant water pressure or any kind of pressure that's in there unless the water was bound in some way. So again, they know that calcium is released out of this capsule first before the tubule everts out. So then they suggested, okay, well, the calcium leaves, and then there's probably some conformational changes of the proteins inside here, or maybe the tubule everts partially, starts to uncoil, or maybe something happens somewhere along within the wall of the nematocyst capsule and squeezes the tubule out that way. So they have all these modifications for different theories, and the bottom line is we don't know exactly what causes a nematocyst to evert, or any specific mechanism in general that will do it, because it can be done through different types of chemicals, and these would be chemicals that the jellyfish would not normally see in its environment. So it's going to be hard to judge like what the actual mechanisms are. Okay, now nudibranchs and sea turtles are jellyfish predators. They eat them. They have ways of dealing with the toxic constituents, either by their way to prevent the nematocyst from discharging, or they have enzymes in their digestive tract that will neutralize the toxins as they eat them. In the case of nudibranchs here, okay, this is a sea slug. It's like the slugs that you get on land without, without shells. They either have a reduced or no shell at all. The difference between these and the land snails is slugs. You don't throw salt on these because they're already in salt water. But these pockets on here, these are called serrata. These outer pocketings on the nudibranchs, they do a peculiar thing. They're used, they don't have gills, but they're used as gas exchange function. But the nudibranchs can sit down and eat a bunch of tentacles off of a sea anemone or off of a jellyfish someplace, and they will selectively retain like the biggest and most virulent nematocysts and incorporate them into their serrata here and use them for their own defensive mechanism. And they don't keep all of them. They just take the ones that'll sting you. So you get theoretically stung by a nudibranch also. And then the sea turtles just eat them. Okay, there's a couple of other organisms that are associated with jellyfish that are apparently not affected by their sting. One is the butterfish or the harvest fish that are found out here. They use the jellyfish bell for protection while they're younger, and then as they get older, they also eat the tentacles themselves and don't get sick from them. And so it would be nice to know that, because if humans could get a handle on what's associated with the discharge mechanism of the nematocyst, then topical products like this can be manufactured, which would be something like a sunscreen that doesn't wash off even when you go in water, and it prevents you from getting stung by the jellyfish. There's no such thing on the market right now, and unfortunately the humans must deal with the aftermath of getting stung by a jellyfish. This isn't that funny because this guy will be lucky if these welts ever go away, first of all. And second of all, he's lucky that he didn't get stung severely enough to lose his life. Okay, finally, that was not here. He was in Australia, where he got stung by that. Australia and in the Indo-Pacific region, that's the box jellyfish. It does that. Okay, I hope this doesn't get over-involved right here, but this is part of the dissertation work that I have done at the University of Maryland, where they have been pursuing jellyfish toxin research for over 20 years. And some people are interested in the bioactive compounds of not only jellyfish, but other marine organisms, because anything that's bioactive might have potential for certain pharmaceuticals and things like that, or they might need it to find the antivenoms for some of these organisms that you could get poisoned by. Okay, so this is just a list of enzymatic activities found in Chrysaor quincacera venom. That is the sea nettle found out here in the Chesapeake Bay. These enzymatic activities have been at least assayed for. They know that they're there. The acid and alkaline proteases proteases in general are enzymes that chew up other proteins. Okay? The acid ones, what is considered an acid protease, is something that works under acidic conditions. Say anything below, since pH 7 is neutral, so we'll say anything below pH 7 below that. If it has optimal activity right there, then it would be considered an acid protease if it chops up certain types of proteins. Now, the proteins that it chops up, it might work specifically or non-specifically at different amino acid residues. Okay, I don't see much use for a jellyfish to have an acid protease unless it's in the stomach contents where digestion might cause like a decrease in the pH right there. In general, the pH of seawater, does anyone know what that is? It's okay. It ranges between 7.5 to 8.0. So it's a basic environment, and this is where acid proteases would be most optimally effective. And these acid proteases might be responsible for, like, the dermonecrosis that that guy in the last slide that you saw on there would get. We don't know exactly that, but there's enough proteases in in the sea nettle venom to possibly be the direct cause for that dermanecrosis. Okay, another enzyme that we have found in the venom, and I hope I can say this without getting tongue-tied, is hyaluronidase. Okay, hyaluronidase is an enzyme that hydrolyzes hyaluronic acid. And hyaluronic acid is a ground substance of connective tissue. Okay, so the hyaluronidase would not be toxic itself, but if it helps to hydrolyze and put little holes within spaces and within your connective tissue, that would be a vector or a route for other toxins to diffuse into and do other damages to other part of the system. It's not then a large quantity in the jellyfish, though, but in comparison to snake venoms have a lot of hyaluronidase in them. And another one is a hemolysin. We found this to be a small peptide, or a small protein that causes hemolysis of your red blood cells, which means if you add them to your red blood cells, it causes all the hemoglobin to leak out. And of course, you don't need that because you need your red cells in order to breed. Okay, the rest of these, which are the non-specific aminoesterases, the ATPase, AMPase, RNase, and DNase, we have found them all in the toxin as constituents of it, but no great research has been done in this area because they probably represent minor constituents of the venom. What isn't on this list that is about the most interesting component of the sea nettle venom is the factor that we call the lethal factor. Okay, the lethal factor is thought to be a large protein molecule that embeds itself into the lipid bilayer of cardiac and nerve cell membranes and causes sodium ions to leak into the cell. And this can be very detrimental, especially with nerve and muscle tissue, because it disrupts the normal resting membrane potentials and causes a cascading effect such as neurotoxicity and cardiotoxicity. And so, like the guy that survived the sting back there, he's lucky. Other people haven't survived. Usually what will happen to them is they'll die due to either cardiac failure or respiratory failure due to the toxic effects of this protein found in the venom. Okay, so my job when I was a graduate student at the University of Delaware at Baltimore was to try and purify this lethal factor. And I devised a purification scheme using, why don't you yell if this is out of focus, it's hard to tell from here, using a couple of conventional protein purification methods. I came up with this scheme to help to identify and isolate the protein that was responsible for lethality, and we tested mice for that. And so don't go, upon IV injection of this toxin will kill mice dead within 30 seconds. Okay, so what you do during all of your purification schemes, if you're purifying proteins, is you make up a table like this, and it'll tell you through each purification step whether you're purifying something or not. What will tell you that is the specific activity that you calculate, and also the purification factor. All of these things are somehow interrelated, so if you get the first line, you can calculate everything else. And so, of course, you want your purification factor to increase as you go along. If you were just dealing with plain enzymes, you'd want your specific activity to increase. It doesn't here because we're dealing with, we're doing LD50s, or lethal dose 50s, or effective concentration 50s. And so it's a concentration of protein. So in this case here, we want our numbers to decrease because the smaller amount of protein needed to kill the mouse, the more pure it's getting. Okay, one other fact you might want to look on is your percent yield. You know that you start out with 100, you try to retain as much as you can, and by the third purification step I only had 3%, but it's better than nothing. Especially when I put my protein samples on gel electrophoresis. This is a technique used a lot by protein chemists to it'll tell you two things. It'll tell you the purity of your sample after you finish your purification procedure, and it will also give you the apparent molecular weight of the substance. So now you can tag something onto it and say, yes, my protein is this big. Okay, and so what this whole thing is a gel made out of an acrylamide some acrylamide polymers, and you apply like your protein samples at the top of the wells. They're way past this picture right now, and the gel's within like 2 plates, okay, 2 glass plates, and then you apply electricity to it, and your proteins migrate going downward, okay? And the smaller proteins move faster, and the larger ones stay more towards the top. So at the end of the run, you take your gel and you stain it for some type of stain that's stained specifically for proteins. In this case, we use silver nitrate. So here is my first purification step. This one was my second purification step. And over here is my third purification step. In this first lane here, I have molecular weight standards. So these are known proteins of known molecular weight, so you have a reference to compare them to. And I found that, that the lethal factor has an apparent molecular weight of 105 kilodaltons. So finally, what you can get from just having a purified protein, whether it's from marine venoms or any other type of protein purification procedures you're doing, and I just recently mentioned this, okay, you can start looking at specific antivenin production, okay? Because we couldn't tell exactly, we think that the lethal factor works as an ionophore or something that pokes holes into the cell membrane and channels sodium ions in, but we can't say that for sure until you have that one protein purified and then you test for it and then you can say, yes, this is what it does. So we still haven't done that yet, but we've got a good indication that, that this is the mode of action of this protein. The other thing is, it's like any type of bioactive compound. If it has biological activity towards any type of cells through modification or lower dosages or whatever, it has a potential for being a new drug, a new pharmaceutical around. And there are some places right now, especially one place in Florida that I know of, they're extracting just about everything out of the ocean they can get in order to look for new antiviral, anti-tumor agents. Okay. Also, purified proteins, especially from natural products, could be used as cellular probes. So even if it kills all the mice, you might be able to use it to either block or activate certain pathways that some cell biologists or molecular biologists want to use in other areas of research. Finally, with the purified protein, you can go ahead and start looking at molecular techniques. I can start working another way now and take the purified protein and start looking and trying to find a sequence of DNA that encodes for this. And if I wanted to mass produce it, I don't know why, but you'd be able to do that. And so that's the end of my talk. And if you have any questions, I'd be happy to try to answer them for you. No, yes.
Might be a silly question, but I've got to ask it. I've heard that meat tenderizer is a good thing to put on a jellyfish steak. Is there any validity to that?
Well, yes.
No, what would you recommend?
Yeah, no, it is. You can use a meat tenderizer because what meat tenderizer is a protease. So it's not going to stop whatever toxin has gone into your skin already. All right. The only thing it's going to do is like get rid of the nematocysts that might still be on your skin. But it's not going to neutralize anything that's like gone in already. Okay, but that meat tenderizer, alcohol, vinegar,